Hello students. Now this is second part of basic molecular biology. We are going to study central dogma. Now this is the central dogma of molecular biology, which includes three processes. Actually four processes: replication, transcription, translation, and another one process which is discovered later is known as reverse transcription. Okay, and this DNA, RNA, and proteins, these are the basic players of molecular biology. Now this DNA, when is duplicated with the help of enzyme DNA polymerase, the process is known as replication. When DNA is transcribed to the RNA with the help of enzyme RNA polymerase, the process is known as transcription. In few organisms, RNA is reverse transcribed and double-stranded DNA is formed. That is, the process is known as reverse transcription. And from RNA, the proteins are synthesized. The process is known as translation. So here there are four processes in central dogma: replication, transcription, reverse transcription, and translation. So this is blue colored arrows which are shown is general process, whereas this red one which is shown, these are special processes. Here the DNA is duplicated, the process is known as replication. From the DNA, RNA is synthesized. All three types of RNAs, mRNA, rRNA and tRNA are synthesized. The process is known as transcription. In few organisms, this RNA is replicated. RNA is used as a template and the DNA is synthesized, double-stranded DNA synthesized. The process is known as reverse transcription. And using RNA, proteins are synthesized. The process is known as translation. Okay, so this is central dogma of molecular biology. This is genetic code. The code is a trinucleotide sequence, that is the triplet. Each triplet codes for one amino acid. The combination of three nucleotide sequence present onto the mRNA dictates one amino acid. Our ribosomes bind to the mRNA, read this triplet codon and drop amino acids one by one, join those amino acids with the help of peptide bond, and polypeptide chain of protein is synthesized, which is further folded so that protein can work. Okay, so this was about central dogma. Next we will start the molecular nature of genetic material. Now the first experiment carried out is by Griffith, Frederick Griffith, in 1928, which is related to the bacterial transformation. Okay? This Frederick Griffith experimented with smooth virulent strains of Streptococcus pneumoniae and rough non-virulent strains of the same organism, Streptococcus pneumoniae. Same organism with two different strains. Okay. He dictated that bacterial transformation demonstrates transfer of genetic material. Next in 1944, Avery, MacLeod and McCarthy, they showed that the DNA is transforming principle. DNA is transforming material. What Griffith discovered? Griffith showed that something is transforming bacteria. Then every MacLeod McCarthy, they showed that the DNA is transforming material. So these are the three major discoveries. Before the DNA structures have been discovered, these are the three major discoveries. Griffith in 1928 demonstrated transformation. Avery MacLeod McCarthy demonstrated that the DNA is transforming principle, and 1952 Hershey Chase, they demonstrated that viral DNA enters the cell. That is, the DNA is transferred from parents to the offspring. Guthrie and Schimmer in 1960 demonstrated viral DNA codes for complete virus. Okay. Then this is Griffith experiment, where he used smooth strains, that is capsulated strains of Streptococcus pneumoniae, which are known as S strain. Smooth stands for S strain, R stands for rough strain, which lacks capsule. Okay. So he first used living smooth strain and injected to the mice. Observed that mouse dies. Then he used rough strains, which are non-capsulated, okay, and injected to the mouse. He found that mouse lives and mouse is healthy. Then he used heat-killed smooth strains and injected to the mouse. Mouse does not die. Mouse lives healthy. Okay. And the next, he mixed both the strains, heat-killed smooth strains and rough strains, and injected to the mouse. He found that mouse died, and later he extracted organisms from the mouse. He found that there are living smooth cells present in mouse body. That is, something has transformed rough strains to the smooth strains. He only discovered that something is transforming non-virulent strains to the virulent strains, but he did not find that what exactly is responsible for transforming this non-virulent rough strains to the virulent smooth strains. Then Avery MacLeod McCarthy did an experiment. What they did is